A very warm welcome to the participants of the Proteomics Advanced Winter School 2021. Presented by Proteomics Lab, Indian Institute of Technology, Bombay. Day 2, Sample Preparation and Gel-Based Proteomics. Lecture 2, Sample Preparation.
I conveyed you the significance, the emphasis about good sample preparation strategies. So as you will appreciate that, proteomics is not just a technique where you can just simply use, a set of protocol just to, do the things like polymerase chain reaction or NGS. It is something which requires really careful considerations about the workflows involved. And that's where you can gather knowledge from the protocols available, different literature review. And I'd suggest you, whenever you are starting your project, it's a good idea to start very unbiased way. Despite you are very knowledgeable in your project, you should still think about what is known in literature, read about them, look at what kind of methods people have used, and see like how best we can learn something from those methodologies and protocols. So I'll be continuing about the sample preparation as we were discussing, and I showed you some workflows involved for doing tissue proteomics based work involving the urea lysis method, and also the bacterial proteomics work involving trizol extraction method. But again, in the lab, whenever we start any new project for a sample which we have not worked on, we start very unbiased way to try out different extraction methods. And that may involve urea lysis or trizol extraction, different way of TC acetone precipitation, or it could be a mixture of some of these. So variety of things we try, and then also look into the publication that from which method, what kind of number of protein people are able to identify. And based on those considerations, then we take one workflow forward. Of course, if you think about proteomics, what we have to see, like, what is the maximum protein which we can obtain from our sample. That is one of the key goal, because then only you are doing something at the proteome wide scale analysis. So that's where, one method cannot be 100% sufficient to provide you all the knowledge, all the glimpse of the entire protein. And what we are talking right now is about global protein analysis. Think about possibilities where you are looking at only phosphoproteome analysis or any type of specific post-translation modification. May I request everybody to mute themselves? Some background is coming. All right, I request everyone to keep themselves muted. So what I'm saying that, whenever you are starting a new project, new protocol, it's good idea to try compare different methods. And sometime when we think about very specific objective like PTM analysis, then your preparation, sample preparation, enrichment strategy is way too different. So we cannot just employ one method and say we can do proteomics using that method for all type of samples and all type of applications. So we have to be very specific for what we are looking into. Plasma proteomics is one of the quite looked after technology and method for doing proteomics, given that plasma represents a lot of protein leaching from all type of tissue to the blood, and then it is very easily accessible from the blood, and it is most commonly being used for all kind of biochemical parameter measurements in the clinical settings. So even a small volume of the leftover plasma can give us a lot of insight about a disease at the proteome level. Also, if you think about if you are trying to look into a potential biomarker for a disease, and if you can identify that biomarker from the plasma sample, then that could be very powerful to get you an insight about that disease, and also it can be clinically applicable because the test could be developed from that. Again, lot of ELISA kits can be made easily looking for the proteins from the plasma sample. So plasma proteomics is one of the very attractive area to investigate.
ठीक है मैम, ये बताइए कि हाँ, all right, everyone.
keep themselves muted. It is possible, I think, given the virtual interaction, all of you have some other engagements as well once in a while. But please do keep yourself muted in the session. So I'll continue with the plasma proteomics, and what I'm going to do now, I will show you briefly some of the steps performed of doing plasma proteomics sample preparation part. So for the plasma proteomics work, as I said, the blood is very commonly collected for all kind of clinical tests, and very small volume of plasma, even leftover, sometimes 200 microliter or 100 microliter, is sufficient for us to do these experiments. What we are showing you, a very small volume of plasma, which can be taken around 15 microliter per se, and you want to deplete the plasma, you want to remove the highly abundant proteins from that. So for doing that, you want to first select that, which column you want to use to deplete your abundant proteins, right? And sometime you have, the columns which can deplete too high abundant proteins, or sometime you can deplete even 12 or 14, abundant proteins. So what we are showing is the 14 abundant protein depletion column which we are using. And now in the resin, the beads which are there for the antibodies, you are mixing your plasma sample and making sure that you are going to keep it sufficient time for incubation, so that the plasma is mixed well with these beads and the slurry what you have. And then you are going to incubate that so that all the abundant proteins like albumin and immunoglobulin, they can bind to these column which contain the antibodies. And then you can do centrifugation so that you can, elute out the unbound protein, which is your low abundant proteins, devoid of albumin and IgGs and other kind of abundant proteins from plasma. So I hope that was brief, but it conveyed you that doing depletion step is very crucial for the plasma analysis. And I must say that plasma is very attractive sample to work with. But given the nature and the cost of these columns, it becomes quite prohibitive because these are commercial kits which comes for these columns. And if you want to do a project of 100 samples, the plasma sample you can easily obtain, but doing the depletion becomes one of the bottleneck. And but if you can afford to do that for large number of samples, that's, I think, a good way to do your proteomics. Now, in case you are not able to do the depletion, what else way you can move forward in your project? So that's where I think we can take a pause and think about some possibilities, some alternate ways. For instance, if you are looking at some very specific regions of the protein which are low abundant, you can just run them on the SDS page and maybe excise that region from the SDS page and do the in-gel digestion of those the pieces of the gel, and try to use that information for doing your in-gel digestion for doing the plasma proteomics. Or let's say you are keen of looking at some specific peptides, you don't want to perform the global proteomics or discovery proteomics. You already know that there are some potential peptide as a biomarker which are highly abundant in the given disease. You are working on, let's say, breast cancer, and you feel like there is a biomarker linked to P53 or EBNA, and you want to identify that using targeted proteomics workflow. So you need not to do depletion in that case. You can just give a shot, try out doing in solution digestion from the plasma sample, and you proceed for looking for specific peptides which you want to monitor in the triple quadrupole mass spec. And there is a good possibility that those peptides you might be able to see that, because over there you're not looking at abundant peptides which are coming from the albumin and IgGs. Whereas if you are doing the discovery work or the global proteomics work, you have to rely on the depletion, because if you run your sample which is mixture of high abundant and low abundant protein, in the global proteomics, the high abundant proteins are going to mask the low abundant protein. So in the mass spec, when you are going to analyze all the peptides in their ionized forms, all the abundant proteins will take over and you will only see the hits as a albumin and IgGs, and sometime your friend creatine will come. So in all of these sample preparation, of course, this sample what I'm showing you, we did recently a project on the COVID. So where we had to be extra cautious about the infection and looking at the nature of these sample in the BCL2 labs. But even otherwise, when you are working on any kind of clinical sample, good idea to make sure that even the sample may not be contaminated, but you may have tuberculosis or something else in that sample. So take extra precaution and biosafety measurements before you process those samples. And even any other biological sample for that matter should be processed with the extra caution and care. And while it is not within the scope of this workshop, but biosafety considerations, performing your study with ethical standards and biosafety is highly required, which I think should be discussed among the participants. And if needed, sometime we can also intervene and give you some clues about how to do that safely. We spent a lot of time recently about just planning for these experiments on the COVID because of the how to do it to the best safety possible manner, and still try to move ahead with these projects. So these will be the kind of challenges which you will face many times, and you have to see like what is the end goal and objective. In the COVID project, we had no idea for what peptides and proteins they are going to look for as for disease severity prognosis. So we had to start from scratch, and that's where we needed to do depletion mandatorily, because otherwise we are going to lose all the low abundant proteins. We have no idea for this disease that what can be potential biomarker. But after we have done these experiments, and now we know these are some set of proteins which are very interesting. Now the targeted validation could be done only on those peptides, and now we can also use the crude plasma, which is undepleted. It is possible some peptides we may not see them if they are low abundant and not enriched sufficient, but most likely we will be able to see most of the peptides. So please keep in mind about weighing your many of these things I'm talking is the real practicality, not just kind of lecture. But the practical issues are very important because your cost is very prohibitive for doing your large number of samples. So discovery work can be done on the maybe smaller set of samples, 30-40 sample, for which you can buy these kits. You can do the depletion, you can perform the discovery experiment nicely, and you have already taken care of all the pre-analytical variables, which I spoke in my previous lecture, and made sure that everything has been stored in immediately, and you process all the samples in a week time. So you have done really everything meticulous way. Now even these 40 sample, if you do depletion, they will give you a very reproducible result. Now we have identified a set of potential candidates which you want to take as a lead forward, then you can come up with a new strategies for validation. You don't need to do depletion for every single thing. Validation strategies could be based on the ELISA based test if you know for abundant protein antibodies available, or it could be based on the targeted proteomics based workflow. So I hope I'm giving you the balanced perspective of how to weigh your options and still move ahead with the cost effective manner to do the large scale proteomics projects. I'll just talk about another sample, which is an interesting sample we had never worked on, nasopharyngeal swab sample, right? And I think most of us had not even heard about this to do proteomics until the COVID came. And recently, because of COVID, when everywhere the nasopharyngeal swab or otopharyngeal swab samples were being collected for real-time PCR analysis, that became a sample of choice for us to think about, can we do proteomics on that? And now for doing this type of project, think about situation like lockdown, extremely labs are all shut down, and we are thinking to work on a sample which is very unsafe, very difficult sample to work with. And these swab samples are being collected in the a tube which contains viral transport medium, VTM. Now this VTM comes from different vendors having different compositions. It also contains sugar, it contains albumin. So now very challenging sample to work with. And this was the real test, how best to do something on this type of sample in a safe manner. So that's where I'll show you, this brief clip of initial processing of the swab, but I can have some discussion about how to take this type of things forward. So now in this situation, the first consideration was before doing any experiment, can we inactivate the virus from the sample, right? And at the same time, we don't want to lose our
audio is missing.
Is that problem for everybody or audio? I am speaking, right? So
no, sir, it's fine actually. Maybe it's for a few people, but it's, your audio is totally fine.
All right, good. So what I'm
Still
showing
no
you
audio.
First nasopharyngeal swab sample, we are keeping them in the heat incubator for incubation so that virus can be inactivated. So as per WHO guideline and some Lancet papers, if you keep these sample at 65 degrees for half an hour, that is safe to work with. And now what we did, we kept the whole assembly, including our trays and including our racks, so that if there's any spillover, anything, it could all be inactivated. I am requesting all the participants, please keep your background muted. Okay, thank you. So this sample, as I said, the swab sample was challenging sample to work with. And first consideration was to inactivate the virus. Actually, we did the same thing with plasma as well on the viral, the COVID patient samples. So once this inactivation was done, then thought was how to get the protein out of it, because, and also we had to think about something which can be clinically applicable. We don't want to go with very long routes. For example, in the urea lysis method, once we go ahead in the next step, I will talk to you that we have to dilute that urea, because that is going to otherwise have problem in the digestion process. So in that way, we thought about take some of the organic solvents and try to precipitate the swab sample and see if we can get sufficient amount of protein from different type of organic solvents. I was talking to you about different precipitation strategies of acetone, TC acetone, variety of things we tried. And we thought to keep it as minimal a step as possible to do this type of work. So I'm just giving you a nutshell here of sample preparation strategies. One for the plasma, which I spoke just now, that we did heat inactivation of the plasma, followed by depletion of the abundant proteins. And then we ran on the gel, SDS-based gel, to look for the how the depletion has worked out as compared to the crude plasma or the undepleted plasma sample. And that looked like quite reduced intensity, although not totally removed, but quite a bit of abundant proteins have been removed. And likewise, we tried the swab sample. And in this case, from the VTM, we tried 3 different methods. Now this one is much more infectious sample, so we did heat treated longer and 65 degrees. And samples are in the acetone, ethanol, or isopropanol, 3 different organic solvents were used. And we had no idea that what is going to work out for us, because of there was no literature available for this type of sample to work with. And so we just tried independently, and thought was, even if a patient is in the remote area, somewhere in the rural area, and you want to collect the sample from the patient, one of the bottleneck was for the real-time PCR work, that the sample has to be collected and stored at the 4 degrees for doing the RNA-based work in the cooling condition. We were thinking peptide being, if you are measuring peptide only, you don't have to amplify the peptide. So as long as we can get the protein precipitates, then one could take that forward and do the digestion to get the peptides. So in that way, whether in the rural areas or for looking for asymptomatic screening, even if the patient, we give patients that, or individuals, these tubes which contains ethanol or isopropanol or acetone, if they can spit, or if their swab can be collected, those protein can be precipitated in the organic solvents, and those could be sent to the facilities to test out. And that was actually, quite attractive strategy, given the nature of the, the screening which is required, and also thinking about the safety of handling this sample. So, so we tried 3 different methods. And after doing these extraction methods and running in the mass spec, we realized that even the viral peptides, some viral peptides are more seen in acetone condition, and some are more seen in ethanol condition, some are more seen in isopropanol condition. So it means if we are looking at nucleoprotein and spike protein, membrane protein, replicase protein, various abundant proteins of virus, and we want to detect virus for detection test, one extraction method may not be best strategy. So and given these are all very cheap organic solvents and very safe to work with, what we thought, precipitate the, take a small volume of swab, precipitate the protein, and after precipitation, then pool that, so that now we enrich the viral peptides, because now we get the more viral peptides coming from this same swab sample from that patient, and then proceed for the urealysis, and followed by do the trypsin digestion and desalting step. So again, you can think about how to come up with the newer methods to work with. For something like this, we had no prior idea, no background, and we thought to explore some new things, so that in the process we can also develop some safe method and quick method to do these type of investigation. And this method actually already being patented. Now we have filed patents for both of these, and also one of the studies just got published. So I'm not talking right now about this work and what we did and what we got out of it, but just giving you a glimpse of it. The different samples require different strategies, and you have to try out new things and newer things to really make some, really insightful idea from that sample. Let's move on to some plant proteomics based sample preparation. This is one of the key area. I think a lot of agriculture scientists, a lot of plant scientists, food people, they all work on plant samples. And but given the diversity of the plant and having variety of interference they have, it makes it attractive but very challenging sample to work with because of the nature of the lysis, which is very harsh lysis required. You have highly abundant proteins like ribisco if you want to work on the plant leaf and want to investigate the effect of infection, what pathogen is infecting a plant leaf. You have to remove those highly abundant proteins like what we did for plasma. So you have to do in that case the ribisco anti-ribisco column you have to use to eliminate those proteins. Likewise, if you're working on the other sample type from plants, you have to remove the chlorophyll, you have to remove the carotenoids, very coloring agents, pigments, or you are looking into the roots. Those are, of course, you have to remove the mud and soil and stuff, but also you have to remove the phenol and other interference which are present in the root. So likewise, you have to think about the good extraction method for doing your plant proteomic sample. But I'll show you one method which works usually well for the plant proteomics type of workflow. So let's watch that demo.
Select the plant leaf sample of interest and weigh around 300 mg of the leaf on an aluminium foil. Transfer these leaves to a chilled mortar and carefully add liquid nitrogen to it, which helps in drying up the leaves instantaneously. Grind the leaves well using a pestle to obtain a fine powder. To this powder, add around 0.5 ml of lysis buffer containing trichloroacetic acid, acetone, and dithiothiotol, and grind it well until a fine paste is obtained. The lysis buffer causes the plant cells to swell and finally break open, thereby disrupting the membrane and releasing all its intracellular contents. Add another 1 ml of lysis buffer to the paste and then transfer the solution into a fresh tube after grinding thoroughly to obtain a uniform mixture. Centrifuge the sample at 10,000 rpm for 5 minutes and maintain a temperature of 4 degrees Celsius during the process to ensure that there is no denaturation of the proteins. Discard the supernatant and retain the pellet containing plant proteins along with various other intracellular components. Incubate the pellet at -20 degrees Celsius for an hour. Protein precipitation. Remove the pellet from -20 degrees Celsius and add chilled acetone to it. Mix the sample well by vortexing to obtain a uniform solution. Centrifuge the contents at 4 degrees Celsius for 5 minutes at a speed of 10,000 rpm. Discard the supernatant and repeat the acetone washing at least 3 times to remove all plant pigments. Obtain protein pellet at room temperature. Reconstitute this pellet with rehydration buffer. vortex the sample to obtain a uniform solution. Centrifuge the contents the following morning at 10,000 rpm for 5 minutes. Collect the supernatant containing proteins in a fresh tube and store it at -20 degrees Celsius until protein quantification is.
All right, so I think we have seen a variety of ways now how to do your different type of sample preparation. During this process, whether you talk about plant or human plasma or human tissue or bacteria or yeast, any of these samples, you will have a variety of interfering substances. For example, salt, you will see it all the time coming from the buffer. And very oftenly seen in the case of urine samples, plasma samples, or even cerebrospinal fluid. Those are highly enriched in the salt, and you have to remove that either by performing dialysis or different type of gel filtration methods, or even when you are doing the TC acetone precipitation, sometime when that removes it very easily, and you can do subsequent washing steps. So many of these are the ways to remove your contaminations, but you are going to see that. And if you are going to plan your two-dimensional electrophoresis type of experiment, this is going to be quite crucial, because your IPG strip or immobilized pH gradient strip, they will stop to process your sample for the isoelectric focusing to happen, because so much voltage will rise and then go to stop, and as a result it can't move in the electric field. So that is one of the important thing to consider if you are planning for the two-dimensional electrophoresis experiment. Or even if you're thinking about directly shotgun proteomics of using LCMS based approach, think about that, you have the nano LC columns in this case, when we are using everything in the very nanoliter volumes. Now, any these kind of contamination can clog your column, and each column is so costly. Like in the mass spectrometer, the major cost comes from the cost of these analytical columns. If they can, if they are getting clogged because of your interfering substance, you're going to lose 1.5 to 2 lakhs rupees cost, just because of your sample was having some interference and some contamination, and could be salt or could be nucleic acid as well. So these things, if you can remove them well and do the desalting step properly, both at the protein level and at a peptide level, I think you are really safeguarding not only your project but also your instruments and your reagents, because these columns and these instruments are quite sensitive to these contaminations. And when you are looking into nucleic acid contamination, sometimes your protein solution which you got is highly viscous, and that's where you can get a sense directly that this sample is probably not the best sample to work with, and you need to do more of the nucleus treatment, DNA dilated mixtures to be incubated and again washed off. Polysaccharides are another challenge. Sometime you will see different glycogen and starch. They are going to be coming along with the protein. They're highly bound. So you have to think about different ways of precipitation and cleanup steps, like ammonium sulfate or using the trichloroacetic acid based precipitation methods. Lipids, again, the different type of tissue sample when we work on the brain tumor tissue or cerebrospinal fluid, variety of these samples are having high lipid enrichment. So delipidation with the organic solvents can be very important in that case. And then of course there are different detergents which you are using, like any detergents. SGS is going to bind very strongly to the complex of the proteins, and that has to be removed before you do your in gel and solution digestion. So again, you have to keep in mind that can you use some of the deuterionic detergents like Chaps, Triton X100. Those could be more useful for you to avoid the contaminations. And of course, as I said, the interference could be very subject to the type of sample which you are working on. There is an endless list to it. Lignins, polyphenols, tannins, alkaloids, pigments, variety of things from the plant samples which are all interfering as well. But one more thing which is crucial here to think about along with all these interfering substance, the way we process our sample, right? Simple things like, you are wearing the gloves all the time and adding some sort of mask and the face shield and small these kind of practices could avoid the keratin contamination, because the things coming from the skin or hair fall, those are the one which we see very prominent and the most abundant peaks in the mass spectrometers. And as a result, you are going to lose out your less abundant proteins. So if you can avoid those contamination, then I think your sample preparation is going to be really good, and you can make use of that for your further mass spec or other proteomics experiments. Now, given that what we are talking is highly complex samples, which is having thousands of protein to investigate. So, of course, problem is complex, so we have to deal with it, right? And let's see how best we can keep the environment cool and background less noise free to proceed to the next step, which is quite complex of handling the samples. So now what we are looking into, thousands of protein which we want to separate. And after all the hard work of extraction, now we have this tube which contains these clean samples with thousands of protein. But if you are going to take that entire proteome and proceed forward for doing your mass spec or other proteome experiment, you might lose the coverage, because proteome coverage will depend upon how best your instrument can really read those peptides and the ions coming from the peptides. So fractionation strategies are another thing to consider while planning your proteomics experiment, how to fractionate my sample. And that's what I was going to show you on the slide, that the different type of sample fractionation strategies are very important. One could even, as I said, even sometime you do not have these columns which you are buying from the commercial ways. You can use some authentic chromatography. You can get rid of the abundant protein from the peaks, and you can also fractionate your sample using electrophoretic method. Of course, we do IEF for the 2D gels, but also you can use off-gel fractionation, which is known as liquid phase IEF. So which is another specialized setup from Agilent, off-gel fractionator, but that's pretty easy to work with, where you can have your protein in the solution form, and you can separate them in the isoelectric focusing unit in the liquid solution form in the cups, and then you can collect, if you have used 3 to 10 pH IPG strip, now that all the cups are for different pH range which you have provided, and you know that between 4 to 5 pH range, these are the cups where you can collect the solution, and likewise you have all the different pH, different proteins have moved into respective PI values. So liquid phase IEF or off-gel fractionator is another way of fractionating your complex sample, also based on the pH value. So you know that these protein fractions are for which pH range. So if you're looking at some more biologically interesting protein of a specific pH range, you're only keen into the basic protein or acidic protein, this could be another interesting way to separate your complex proteome and fractionate that, and only run those fractions where you have maximum interest to look into your proteins of interest. Of course, conventionally people use a variety of other methods, including differential centrifugation methods, sequential extraction methods. All of them could really make different layers, and you can separate your complex proteome mixture into different fractions which can be taken forward for subsequent analysis. I think in the interactive session somebody mentioned about the working on the algal samples, and so I just thought to show this slide that this is a kind of project which we had not worked very intensely in the lab. This is a nanochloropsis sample, and again, when you do not have good idea for the sample which you are working with, good idea to try out different things, so that like which method is going to work out well. So we had not worked on the nanochloropsis, so we tried out triazole extraction method, phenol method, TC acetone method, and very interestingly and surprisingly, we found that each of these methods are giving some unique number of proteins. All three of them are giving substantial number of unique proteins from each methodology. And the way each extraction method works, some is going to enrich your membrane proteins, some are going to remove lot of bound proteins, some may have done the certain bound fraction, and likewise you are going to enrich sometime different transcription factors or kinases. So then you are going to have differential expression of different proteins and enrichment if you try different extraction method. So that you cannot use just a logic that is what I'm going to use. You have to try out different things and see where you can get your most of the proteins possible. So by looking into this type of data, and just recently we are doing a project on the breast cancer samples, and we are trying to optimize that what can be the best condition possible, and results are something very similar. When we are trying 4 or 5 different methods, we can see that there are a set of unique proteins which can be enriched from a unique method. And therefore, if we can afford our time and effort to analyze the sample separately, or at least extract the protein separately from each method, then that is going to enrich our proteome. So many times, if you think that you want to claim for that you want to do a proteome-wide investigation, so your goal is to be as many protein as possible, right? For example, you read Professor Matthias Munn's paper, and the goal over there will be to reach out to closer to the whole proteome in single shot run, right? They showed very nice papers on the yeast, for example, in single shot 2 hour, 3 hour of injection with same sample unfractionated, you can totally try to map the whole proteome. And likewise, so your goal ideally should be to map the whole proteome from a given sample type. So if that is the goal, then you'd like to employ different extraction methods so that you can enrich the protein from different methods. Then you can pool those proteins and make it one pool, which now contains the common, different unique proteins as well as common from all the methods, and use that pool of the sample to take forward for your further downstream analysis for proteomics work. And something similar we did recently in the nasopharyngeal swab sample of the COVID patients as well. Just to summarize the first part of this presentation from the previous lecture and continuing now, is that the sample prep is of course most crucial step for doing proteomics. It is not only just you have to know how to process your protein extraction method, but also you have to think about which technology you're going to take forward for which type of question to be addressed. Accordingly, you are going to enrich your sample and you're going to fractionate your sample. You're going to remove the contaminants from the sample, keeping that objective in mind. And as I said, it's good idea to think about all the biochemistry what you have studied, be critical, be intellectual, be creative, and think about, read literature, but also be open to new methods which you can try yourself. Sometime your 6 months of investment of just trying out new methods can give you, it will be a frustrating journey to explore many things for 6 months. But after that, once you have reached to the deep proteome coverage with a good reproducibility, then your projects will start flying very fast. And in 3 months, you can generate, publication quality, high quality data sets very easily. So that's something which I'd like to emphasize from the, the sample preparation part. While in the entire proteomics, there are 3 major components. One is the sample preparation part. Second is the instrumentation part. And third is the data analysis part. Most of the time, most of the people feel that the middle part, the instrumentation part, is most crucial. And you always feel that you may not have access to the best technologies and best instruments, so you should not do proteomics. But I think that's where there is a gap here in understanding. We should be thinking about only the first part and the third part more importantly, or most importantly, because the sample preparation, if you can make a good sample, you can send the sample to different places and everybody will generate good data quality. And third part is data analysis, which you should be pick up from this course, how to analyze the data yourself. And if you can analyze the data yourself, then I think you can do justice to your sample, because only you know your sample, you know your interference, you know the contamination, you know what is the artifact, what is the real signal, what is the correct database to be selected. All that cannot come from anybody, any facility, or any other collaborator for that matter. You have to do that yourself. So first part and third part, sample prep, making good sample, and third part is data analysis, are the most crucial in the entire proteomics workflow, in my own personal opinion. Middle part looks very gadget oriented and the cost oriented, but is actually the simplest part, because if instrument is good, facility is good, technicians are good, they will generate good quality data irrespective of different projects. But the sample intake has to be really crucial for that matter. So just to continuing on this, on the sample prep part, before we go to the more interactive sessions. Now you have identified that this extraction methods work very well for your sample. You want to know how much protein you got from each of the method type and what is the protein quantification yield from those methods, and then how the quality of these samples look like visually and also quantification wise. So Bradford assay is one of the commonly used methods. So again, given that all of you are at quite senior level, I don't want to make it again a basic course of my classes. So just to refresh you briefly here, but I'd like to show you more hands-on. So you are using different type of assays for doing quantification. Please use that. There is no need to only shift to Bradford. So I just want to convey one thing, that there is no quantification method which is the best method. You can choose the quantification method the way you think is working well for your lab. Could be Lorry method, could be any other kit which you are using, BCA method. Anything is actually good, and none of them are actually perfect, right? They're all subjective. They give you some landmarking and some idea that okay, your protein sample is 100 microgram is around this, or this sample is 150 microgram. So they are giving you some relative idea, but they cannot be absolute that it is exactly 100 microgram, right? So that's where I think you can choose to select your own method for quantification. We are just giving you one for illustration and discussion purpose. So Kumasi Brilliant Blue based method, Bradford assays and modified Bradford assays are quite popular as well. And you are measuring the binding absorption max shift from the 465 nanometer to 595 nanometer. When the protein binds, the red color dye change to the blue color form because of the hydrophobic interaction with the positively charged amino acids. And that's what you are measuring at the 595 nanometer. For doing that, we make the standards with the known protein like BSA. And again, some of these things could, please don't feel that we are trying to teach you something very new. You are already doing it. We are just giving you some recap and also giving you an idea that how you should be careful about some of these steps as you go along. So you should make some standard graph the way you have doing for other protein assays, and choose the any standard protein and make the known amount of those protein, their stocks, and measure their absorbance, and do the plotting, make the standard curve for the known amount of, let's say, BSA in this case, and the absorbance value measured at 595 nanometer. Calculate the R square value, and the graph should be as linear as possible with the good R square value, in this case almost 0.99, which is good. What we are showing you here, some actual data for some samples. So take at least 2 absorbance for every sample, because you will find sometimes some variability. But if at least you should take the average of those. If you find one is very different than other, you should repeat one more time or 2 more times, because your protein quantification is very important to start with. And then depending on how much volume you have taken for quantification, you can make it per microliter, and then choose accordingly that how many microliter you need for different application, for 50 microgram, 30 microgram, 5 microgram, likewise. Let me show you this clip of demonstration for the Bradford assay from the lab.
In order to know the protein concentration in a given sample, or to get the total yield of protein in a given sample, it should be quantified. Depending upon the type of lysis buffer used, various quantification method can be used. Today, we will demonstrate you one such method, which is Bradford protein assay. Bradford protein is one of the common method used for protein quantification, and it is a colorimetric assay. Before performing the assay, firstly we should have a standard protein. The standard protein here we are using is bovine serum albumin, that is BSA. So here we have prepared serial dilutions of BSA, that is standard 1 to standard 5, and also one is blank, which will involve only the reagent. And here we have one unknown sample. Now to each of the tube, equal volume of Bradford reagent is added. After addition of reagent, now the tubes should be kept in dark for 5 to 10 minutes. Bradford reagent exists in 2 different forms. One, when it is unbound, it is in brown color, and then when it binds to protein, it becomes blue color. So the intensity of blue color is directly proportional to the amount of protein present in the given sample. It is basically based on the absorbance shift, which unbound form to bound form changes from 465 nanometer to 595 nanometer. So absorbance should be taken at 595 nanometer. Now after incubation, each sample is added to the plate. Preferably the sample should be added in duplicates. Now all the samples are added in the plate, and then it is ready for taking the readings. For this, we are using Multiscan Geo. Plate reader. The first step is to set the plate layout, which includes blanks and all the standards. Also the unknown sample. The next is protocol, in which absorbance at 595 is set. Measurement mode is selected to be precision. And also blank subtraction is set. After getting the absorbance values, the values can be exported. So here we have the absorbance values for all the standards, including standard 1 to 5, as well as the unknown sample 1. So here we have absorbance 1, that is OD1 at 595, and OD2. So firstly, we will calculate the average absorbance. For all the sample, average absorbance is calculated. Similarly, for the unknown sample, average absorbance is calculated. So next step is to plot the standard curve for BSA. For this, sample amount and average OD to be taken. So here we have the plot for standard. Also the R square and equation is displayed, and we can see that we have got R square of 0.99, which means that we can use this equation for the quantification of unknown sample. Using the equation obtained, amount of the protein in the unknown sample can be calculated. So for the unknown sample, we have to calculate X, and the absorbance obtained for the unknown sample would be as Y, as shown in the given formula. Since we have taken 2 microliter of unknown sample, so concentration would be half of it. Now we have got the concentration of our unknown sample. This is how we can get the protein quantified. Its concentration as well as yield can be calculated. Other than the Bradford method, there are several other methods such as BCA and 2D quant. So after getting the protein quantification, next procedure can be performed, such as SDS page or any other proteomic procedure.
All right, so once we now know that we got good yield of protein and we have sufficient amount of protein from the given sample, then we would like to check the quality, that how good the quality of the protein is on the SDS page gel. And many times you might feel that from the quantification you have very high values and you think you got very good yield, but actually in reality you may not have so much good yield. And we will talk more about the SDS page and gels tomorrow, but I'll just show this now lab demo about how to check the quality of your samples on SDS page gel.
Quality of the protein in a given sample can be checked using a gel. For this, one-dimensional SDS page can be used. Here we will demonstrate you how to cast SDS page and how to perform the gel electrophoresis. For casting the gel, we need the casting apparatus and glass plates. The glass plates, one is thick plate, another is thin plate, and suitable sized comb is also required. Glass plates are fitted into the casting apparatus so that the space created between the thin plate and the thick plate can be poured with the gel solutions. For performing the discontinuous type of SDS page, 2 different gel solutions are required, which is one is resolving gel solution and another is stacking gel solution. For these 2 solutions, composition is different, such as this could be the composition of resolving gel and stacking gel. So the first step is to add the resolving gel solution inside the space between the plates and let it be solidified. After some time, now we can see that resolving gel is casted. Next we have to add the stacking gel solution. Now the stacking gel is added. Because we need to create wells inside it, now we will add the comb into the solution. Let it for solidification for some time. Now we can see the gel is casted. It is ready for performing electrophoresis. Now the casted gel is placed inside the electrophoretic tank along with SDS running buffer. Comb is removed to expose the wells so that the sample can be loaded. Suitable amount of protein is loaded for checking the quality. The sample can be loaded in duplicates along with protein ladder so that the molecular weight can be estimated for the proteins. Now we can see that the sample is loaded and it is ready to perform the electrophoresis. After setting the suitable voltage, start running. Here we can see that electrophoresis has started. It has to be performed for around 1 to 2 hours. So the gel electrophoresis may take around 2 hours, and after that, in order to visualize the proteins, it should be stained and de-stained to remove the extra stain. As the sample run would take some time to complete the electrophoresis, we are showing one of the demo gel to show how the samples look after electrophoresis. As we can see here, that clear bands in each sample are seen, which shows that the quality of the sample is good to take forward. So this is how to check the quality of the sample and to decide whether to take it forward or not. After getting the quality check, so sample can be processed for further downstream processing, which can be either gel based or in a gel free manner.
All right. So I think this kind of image speaks a lot about samples, right? SDS page image, when you see something like this, well, it shows that all the proteins of low and high molecular weight, they're very well resolved in the gel. So the extraction method has worked out well. But if all these samples we are loading, let's say 10 microgram of protein in all the wells, then I think we have somewhere wrongly quantified our protein, misquantified. So again, the gels can give you a lot of information after looking at the images, and you can a lot of sense from that to see, like, is that really good for taking forward, or you have to redo extraction method or redo the quantification. As I said, I can make a lot of observation here that some of these extraction methods that work very well, but this quantification is not good. This quantification is not good. There is no protein here. So either the quantification is poor or this method has not worked. So accordingly, we can either re-quantify or try out newer methods as well. So quality control checks are very crucial to make sense whether we are going ahead or we should take a step back and just try out new things. I think in the sample preparation part, if you are having a lot of patience and you can keep taking the backward step couple of times, you will do good justice to your projects and to yourself for doing good proteomics work, because more and more time and effort you put in for this step, you'll be better prepared with your samples for the downstream for the analysis part. So this is one of the way I think you should be looking at your gels. But also I would say that many times you're not sure whether your protein may contain some nucleic acid contamination also. So same protein solution could also be run on the agarose gel, and that is not something which you will do. Otherwise, you will only run the nucleic acid on the agarose gel electrophoresis. But if you run this sample also, protein sample on agarose gel, and you see there are some smear or there are some contamination coming from the nucleic acid trace, then you know that okay, this could be what value you see in the quantification. That may not be truly just because of the protein. Sometimes even the nucleic acid can also be binding to these dyes, and they are giving you some artifactual values, which apparently looks very high to you, and you feel you got too much protein on it, but you may not have sufficient protein from the given sample. So doing a QC check of the nucleic acid contamination is of course important. You can always say that okay, I have used some nucleases and that has been removed, then that is fine, then you are sure about it. So these are the things which looks very simple, which every lab does. But I think when you are thinking about high throughput experiments and high gadget instruments, before proceeding those, we have to do a lot of work into this part to make our samples really good, to take to the next level of analysis for mass spec or other type of proteomics workflow. So once you think that you have got good protein yield, like you have good concentration of the protein, sufficient amount of protein, you got 200, 300 microgram protein you have to at least to work with. So now you want to go for the next step and you want to find out what should be my next step for the proteomics. So for doing that, can we think about what is our goal? Are we going to do the gel based or are we going to do gel free proteomics? So again, for each one of those methods, there is a different way to move forward. If we are thinking about just the simple protein identification from the SDS page, then that should be done with the in-gel digestion. But if our goal was to look at identifying a large number of protein and their quantification, then that should be with the in-solution digestion. Again, we'll talk about more about going for in-gel or in-solution digestion when we go to the next steps of for the mass spectrometers. But right now, I want to just make you prepared to think about a sample prep. But when I said it is just not only about protein extraction, it is starting from knowing your sample type, interferences in that, quantifying the protein, quality control checks, and also then proceeding for the in-gel or in-solution digestion. So let me now show you one lab demo for in-solution digestion.
For the mass spectrometric analysis, in order to identify the proteins in a sample, peptides are required. For getting the peptides, the protein sample should be first digested. Protein digestion can be performed in a gel-free or a gel-based manner. We will show you how to perform the in-solution digestion, that is gel-free procedure. So now, after quantification, suitable volume of the sample is taken for digestion. Before doing the actual trypsin digestion, we need to process through the two steps, that is reduction and alkylation of proteins. For reduction, here we are using TCEP, that is tris carboxyethyl phosphine. Appropriate volume of TCEP is added. The sample is properly vortexed and spun. After addition of TCEP, that is the reducing agent, it should be incubated at 37 degrees for 1 hour. So after getting incubated at 37 degrees, the samples would be reduced. So after incubation at 37 degrees, reduction is happening. That means all the disulfide bonds are broken within the proteins. So now it's time to alkylate the sample. For alkylation, iodoacetamide is used. During the alkylation procedure, alkylation will block the thiol groups so that the disulfide should not be reformed. After this, the sample is properly vortexed and spun. To perform the efficient alkylation, the sample should be kept in dark for 10 to 15 minutes. So after completing the steps of reduction and alkylation, the next step is addition of trypsin for the digestion. So before that, the step is dilution, which means we first need to dilute the molarity of urea that was present in the initial sample. So the diluent is used for diluting the sample. Appropriate volume of diluent is added to the sample in order to dilute it. Before addition of trypsin, make sure that the pH is 8 so that it can work properly. Now the next step is the addition of trypsin that could be added in the ratio of 1:30 to 1:50, which means that 1 part of the trypsin can be sufficient for 30 parts of protein. The sample should be vortexed properly and spun. And finally, for optimum protein digestion, the samples need to be incubated at 37 degrees for around 16 to 17 hours. After 16 to 17 hours of incubation, the protein sample is completely digested, and then the sample is dried in order to get the peptide sample. These dried peptides can be taken further for mass spectrometry. For mass spectrometry, the peptide sample should be properly cleaned. To make sure that the sample is clean of various salts or other contaminants, the sample can be performed through the cleanup steps. For these steps, we use C18 columns. So these columns can be made using the C18 material within the tips. And then the sample is passed several times through the column. This column works on the principle of reverse phase chromatography. During several steps of passing the sample through the column, the peptides would be bound to the column, and then finally the peptide would be eluted. After the peptides are bound to the C18 column, they can be further eluted using different gradients of acetonitrile. And then the elutes is again vacuum dried, and the sample can be taken for mass spectrometry.
All right. So I think in solution is quite important step. And what we are giving you is some generic methods or one representative method to say you always need not only use trypsin enzyme, you can always use a mixture of proteases you can use for the chymotrypsin or lyse. And also what trypsin we have just mentioned or used is commonly used trypsin. If you use some very high quality trypsin or the very fast trypsin, which are more costly, they can also be used with 1 hour or 4 hours of digestion. One could achieve sufficient digestion as well. So there are different products which we have procured recently to try out, and those could give you the digestion in efficiency in very short time as well. So if the goal was to develop some sort of detection test, something what we were thinking for COVID, that's where I think the high efficiency enzymes with the fast digestion can be useful. But in general, I think in the biological experiments, you have sufficient time. You can set up a reaction overnight, and next day you can take it out, and that gives usually a good trypsin digestion uniformly with the overnight incubation conditions. So just to summarize my the 2 lectures. Essentially, many people have done genomics in different labs at different education level by doing DNA extraction and doing PCR and doing basic molecular biology. And people actually expect the same from proteomics as well. And that's why many times they feel like a facility or somebody else as a collaborator has not done great job for their proteomics project. But what is now you are probably getting a sense that there are many considerations required to do good proteomics experiment. And you have to really optimize those things at different stages. So it's much more challenging than the genomics. And you have to be really on top of literature review to understand the methodologies have gone into developing those protocols, and then choose carefully about what you can do within the constraints of your lab and reagents and many things possibilities from your sample type. Now QC checks, as I said, the quality control steps are very important, and that should be done after every step, so that you know that where you are getting stuck, where you don't have good yield, you don't have good quality of the protein, you are seeing the degradation products are happening, you don't see much proteins coming out from the column, or you don't see the peptides concentration is very weak after in solution digestion. So after every step, if you can do the QC checks, then you don't have to inject every sample to mass spec and you say your the project did not come good, because you have already troubleshooted at each step. So finally you are going to get some good results at the end for the actual experiments. So please do keep in many of these things in mind for doing good experiments. And again, the considerations will be very different when you are thinking about doing a 2D electrophoretic based experiment or directly shotgun based LC-MS based experiment. Whether you are thinking about a label free quantification or you are thinking about label based quantification, many things can change, and accordingly your strategies for doing your sample preparation for in-gel digestion or in solution digestion will also change. We will talk more about the in solution and in-gel digestion in the subsequent module when we talk about mass spec workflow, and also that how to make sure that you are getting good quality of the peptides and their quantification prior to mass spectrometry based injection. But I hope given the complexity of the field of proteomics, at least I was able to give you some flavor of the challenges involved, and there is no set recipe which I can give you that these are 1 to 10 steps which you can use for every project and everybody should be happy to do proteomics. That is not possible. We can be engaged in more intellectual discussions about how to make your type of sample and work forward for doing complex proteome analysis. And that's where I think a lot of discussions, lot of reading, lot of thoughts are required to do that kind of experiments. That's pretty much it for this lecture. I will stop by this presentation right now. What we thought in the